Hi everyone, welcome to our HSC Biology Head Start January lecture series. I hope you guys are all doing really well, having a wonderful time in these holidays and an amazing start to the year. So without further ado, let us have a look at today's lecture. So once again, a very warm welcome to everyone from all of us here at 8 Hour Notes. Yeah, lovely to be back for another lecture series. I hope you guys are excited to be back for another lecture series. And today we are looking at module 5 review and a module 6 head start. So before we begin, I'll just quickly go through some of the amazing resources that we have to offer here at 8 Notes that you can access for free on our website, including our study notes. So you can get access to hundreds of downloadable study resources that have been produced by your peers from around New South Wales for the HSC, and you'll be able to get access to those. You can post your own notes up, and it's a wonderful community of like-minded students helping each other out. We obviously have our lectures on this week, so attend as many lectures as those that are relevant for you. Make the most out of it. It's free. It's a great opportunity for you to get a bit of a head start on the content, get a foundational understanding of some of the concepts, and ask lots and lots of questions. No question's a silly question. And we are live on the chats helping you guys out with any questions, so please feel free to ask. We also have our online discussion forums, again, students and tutors, so some of us are on there as well, and again it's a great opportunity for you to get in touch with like-minded people, ask questions from fellow students, from us tutors if you have any questions about uni or anything else that we can help out with. And it's an amazing community. We also have our videos, newsletters, and articles, and our articles usually revolve around like study tips and strategies. To check those out, and check out all the other resources on our website. So along with that, we also have 8 Notes Plus, which is a paid resource, but it is our very own, basically a catalog of a variety of resources, including our study guides, and which includes our topic test books. It includes our notes, also includes our text guides and NEEP past papers as well, exam papers, along with flashcards. So there are flashcards available for a variety of subjects, and we also have these lecture videos that are also now available on the platform. So so definitely check it out. And we also have our own AI answer bot that takes information from our books and our content and is able to answer your questions. So let's get started. Now, before I introduce myself, what we're going to do is I am going to introduce you to a couple of housekeeping rules. So first of all, I just want to make sure that just a couple of things that I remember to tell you. Firstly, this is a recorded session. So this is a recorded session, but I am live answering your questions in the chat. So please ask however many questions as you want to ask. There are, no question's a silly question. I'm here to help, so please don't hesitate to ask. And you'll also find the recording of this session available for a week after the session has finished, and then after that it is available on 8 Notes Plus. But you will have access to the slides that you can download just below this page where I am speaking. But again, if you have any questions, please don't hesitate to ask in the chat. So my name is Aditi. I graduated in 2021, which is crazy because it's been almost 3 years now. don't want to think about it. But yeah, 2021, with an 8 out of 95, and the subjects that I teach include English Extension, Advanced, PDHPE, and Biology. And I am currently at the University of Sydney studying a Bachelor of Science and Bachelor of Advanced Studies in Medical Science as a Dal scholar. And generally love reading, good shows, sunny days, a huge Potterhead, love my classics, especially Austen. Alrighty, so that's kind of it. So let's have a look firstly at Module 5. So like I said, I am live answering your questions, so please feel free to ask any questions. And today we are going to do a Module 5 review quickly first, and then we are going to move on to Module 6. So this is what the scope of the HSC syllabus looks like for biology. As you know, there are 4 modules. The first one is heredity, followed by genetic change, then infectious diseases, and we finish off with non-infectious diseases. Now, you may not study them in this specific order, and that's totally okay. What we are going to do today is we are sort of going order-wise. So in the last lecture series, which was in October, we went through Module 5 and we looked at heredity. We talked about, we talked and covered the following aspects of the syllabus: so reproduction, cell replication, DNA and polypeptide synthesis, genetic variation, and inheritance patterns in a population. So we covered that last time in the last lecture series. In this lecture series, we are moving forward with Module 6, genetic change. So we are going to look at mutations, we are going to talk about biotechnology, and also genetic technologies. So let's firstly begin though by reviewing Module 5. I am assuming, and I'll ask this in the chat, I'm assuming that most of you are probably round about Module 6, because most schools do tend to do them in that specific order of 5, 6, 7, 8. However, if not, if this is new content that we're just going through now, just take it in, ask questions, and yeah, just try to grasp some foundational concepts if you haven't come across them yet. And like I said, if anything doesn't make sense, please ask. That's what I'm here for. So let's have a look. Firstly, so the first dot point is reproduction, and our inquiry question is: how does reproduction ensure the continuity of species? This is basically what everything comes down to, the continuity of species. Really, really important. It's a really important concept, and I just want to make sure that I highlight that. So with every, essentially with every dot point, with every dot point, for example, like reproduction and then genetic variation and so on, they give you an inquiry question, and essentially your aim should be to go through all the points that are listed underneath the syllabus, get all the information, and then once you are done, you should be able to come back to answer this inquiry question. This is the big question that you should be able to answer once you've studied everything under a certain dot point. So a really good way to test yourself is to basically phrase this as a question, and then this could probably be like a 9 marker or something, and then you go under and then you go through every little or rather with every you go through every dot point underneath this main question. So in terms of Module 5, right, so this is, remember, again, a quick summary. A quick summary. So we've got reproduction, we then look at cell replication, DNA polypeptide synthesis. So it's like we start off with a big concept, we look at the smaller sort of part of it, how does it actually happen, what is the relevance of it, and again, how does it happen, and then we link it to the bigger picture here. So we are looking at how traits are passed from a generation to a generation on a system, cellular, and molecular level. So this is, so we look at these system levels, and then we look at a cellular and a molecular level. So remember, we are considering how genetic information stored as DNA is expressed as external traits, and then we look at how does variation actually happen in populations, and then how we can trace this variation within populations to gather data and to then act upon that data. So let's talk about firstly reproduction. So you looked at animal So you looked at external and internal fertilization animals, would have looked at asexual reproduction, which includes vegetative propagation and apomixis. Oops, and apomixis in plants, and also sexual reproduction. So you should essentially know. Apologies, I've just got a bit of a sore throat, so if I cough and sneeze in this lecture, please excuse me. So I was just saying, for example, with animals where you looked at internal and external fertilization, you should obviously know what the process is, and you should be able to talk about the advantages and disadvantages of each. Same thing with plants, and you want to know examples, for example, of plants that of like more popular methods of asexual reproduction versus sexual reproduction and how that occurs. You would have looked at fungi, and within that talked specifically about the processes of budding, spores, and fragmentation in asexual reproduction, and you would have also looked at sexual reproduction. Protists, so be able to talk about the processes of binary fission and budding as forms of asexual reproduction, and also the method for sexual reproduction. And finally, in bacteria, the only method of reproduction is binary fission, which is asexual. So in terms of looking at asexual versus sexual reproduction and looking at some advantages versus disadvantages, just a very brief summary. Remember, it's a very quick review of module 5. Unfortunately, I don't have time to go through all of it in a lot of depth. We did that in the last lecture series, so maybe check that out. But yeah, I'll just quickly go through some advantages and disadvantages very quickly. So the advantages of asexual reproduction are that it's quick, there isn't a requirement for mates, it isn't an energy intensive process, nor is there a requirement to care for the offspring. The advantages of sexual reproduction is that it leads to the production of new alleles, which then also contributes to variation, and it gives individuals the ability to adapt to their environment and hence aid in natural selection. Disadvantages of asexual reproduction are that firstly, clones, because it leads to the production of clones, there is a lack of diversity. It also leads to reduced adaptive abilities, and individuals are more susceptible to large-scale extinction events because of that lack of variation. The disadvantages of sexual reproduction is that it is time intensive, it is energy intensive, it does require mating, and it also leads to fewer offspring. So it's kind of like the opposite of the advantages of asexual reproduction. Okay, just give me 1 minute to grab some tissues and I'll be back. Alright, I'm back. Apologies for that. Okay, here we go. Now let's have a look at reproduction in mammals. So this is where we would look at the process of fertilization and implantation. You want to know what those processes are, and you want to know the structures that are involved. So fertilization is the fusion of gametes to initiate the development of a new organism, right? And it takes about 12 to 24 hours, and this is where fertilization is the fusion of the egg and the sperm. Implantation is when the fertilized egg actually adheres to the wall of the uterus, and when it adheres to the wall of the uterus, we then start calling it a blastocyst. And the process of hormonal control in this whole process is looking at the role of the endocrine system in basically controlling the whole process of pregnancy. So this is where this information comes into use. We essentially use this knowledge of fertilization, implantation, and the role of hormones in this whole process, and the significance of these hormones, to then manipulate this process. And by manipulating this process, we can essentially it can essentially lead to desired outcomes that we want, and that can have a huge social effect and a huge social and economical impact as well. And this is used in selective breeding, in a process called selective breeding, where we essentially apply this knowledge. So what we do is that we know that in order for pregnancy to occur, the sperm needs to encounter the egg at a certain time. And what we do in order to then achieve that is that we time breeding so that it occurs during the intersecting fertility cycles. And we do that because we know that the phenotypic traits, for example, of two different organisms are heritable, so it will be passed on to the offspring. And so to influence the traits, of the livestock offspring, we either crossbreed or we purebreed. And this has actually led to the development of a lot of new species, including the Jersey or Angus cows. And there we have a photo. So this is essentially the two big parts of this first dot point of reproduction. We look at asexual versus sexual reproduction in a range of organisms, in a range of species, and then we essentially look at the process of fertilization, implantation in mammals, and we look at how that can be manipulated to achieve a desired effect, and we can see that in the example of selective breeding. So now let's move on and let's have a look at cell replication. So how important is it for our genetic material to be replicated exactly? Again, this is a question you should be able to answer once we have reviewed this content. So here we are, a couple of key structures to remember before we even begin. So these are chromosomes. Yep, both of them are chromosomes. Chromosomes will have a nuclear envelope, and there will be a mitotic spindle involved, and obviously the cell has a cell membrane. So this is a chromosome, and so is this, and this is also one chromosome and one chromosome. So as you can see, they're both homologous chromosomes, and they both have a centromere in the middle, this little round thing here. However, these are also chromosomes, right? Still counts as one chromosome, even though it's got two parts now. And the reason it has two parts is because it has replicated. So it has a sister chromatid. So the second bit here is a sister chromatid, but it still has the centromere in the middle, as you can see, centromere in the middle, and then the sister chromatid. So in this process, you want to know how many rounds of division occur. You want to know the number of daughter cells that we end up with. You want to know the chromosome number of the daughter cells, how many chromosomes, and you also want to know the role of this process in the body. So here we are. This is our process. So we have mitosis right there, and we have meiosis. So what happens is, firstly, in mitosis, we begin with interphase. This is our nuclear envelope here. In the nucleus, this is a nucleolus in the middle. This is our cell, and this is the mitotic spindle there. Interphase. In interphase, the DNA has already been replicated. That's the key to interphase. The DNA has been replicated, because remember, if the DNA hasn't been replicated, then we only would have one of these. So if we only have one of these, how are we supposed to pull them apart? That's why the DNA has been replicated, and we have these homologous chromosomes with two sister chromatids right there. So DNA has been replicated in interphase, and it's ready to go. And then in prophase, what happens is that our mitotic spindles, they start to stretch. They're almost like these spider-like projections, basically, and it starts to stretch, and the nuclear envelope disintegrates, or so that we can get access to our chromosomes right there. Once that happens, we have metaphase. In metaphase, these chromosomes line up. We do have a process in the middle called prometaphase, but don't stress too much about it. So interphase, prophase, just metaphase them, and we've got our chromosomes lining up in the equator of the cell, and now we've got the mitotic spindle going ahead and attaching to either side of the chromatids here. And once the mitotic spindle has done that, in anaphase, they're going to be pulled away from each other. So you can remember anaphase, they're pulled away from each other. From there, in telophase, what happens is once those chromosomes are pulled apart, and now we have a copy of the chromosome on each side, we then start to see the nuclear envelope form again right there. It forms again, and then the cell begins to essentially not duplicate, but it begins to stretch, so that now we've got the beginnings of the two cells here. That's telophase and cytokinesis, which isn't technically a part of it because we usually say it's interphase, prophase, metaphase, anaphase, and telophase, but cytokinesis is a part of it because it's in cytokinesis that the cell wall is reinstated, and now we've got two cells with the chromosomes, with the same number of chromosomes. And the key here is that mitosis is a process that occurs in the body cells. So we essentially end up with the same with the same number, and also the same sort of chromosomes, the same chromosomes in both of them. So the genetic information is the same. The genetic information does not change here. Yep, so that's the same. This process occurs in the somatic cells of the body, so our body cells. Every time we injure ourselves and we need new cells, this process will occur. Every time the cells die, this process will occur. So that's mitosis. Meiosis, on the other hand, is a process that affects only the gametes. So this is the process that occurs to form the gametes, to form the sex cells. And the process is essentially the same, so interphase, prophase, metaphase, anaphase, telophase, and cytokinesis, but it actually happens twice. So that's why we separate into meiosis 1 and meiosis 2. Meiosis 1 is when we essentially put a 1 in front of each of these initial processes, and then mitosis 2, we put a 2 in front of each. We again begin with interphase, where our DNA has been replicated, right? DNA is copied, and then we go into prophase. In prophase, a really cool step occurs. The chromosome pairs line up, and just as they line up and they are to be separated at the equator, as the chromosomes pair up, what they do is when they pair up, they exchange genetic information. So they're homologous chromosomes, which means they share similar characteristics, and so therefore, for example, if on one chromosome, they're going to pair up, and both chromosomes, let's say, at the same spot in the chromosome, they have let's say alleles for eye color. What they're going to do is they're going to swap over that information. So let's say the chromosome that initially may had an allele for brown eye color may have swapped with the allele for blue eye color, and then they swap over, and that leads to variation because it's a recombination. A new thing is occurring here. A new combination is being created here. So that is prophase. It's a very cool process of crossing over, and it is usually tested quite a bit because they want you to know how genetic variation arises from this crossing over. So that was prophase, and then we've got metaphase. This is where the chromosomes all line up. Again, we've got a mitotic spindle which has these spider-like projections that are going to hold on to either side of the chromosomes. In anaphase, now look here, as you can see, we can see that the color here has switched, and this is just to show you visually that they have exchanged information. That's all that is. But anyways, anaphase, the chromosomes are pulled apart, and then we see telophase 1 and cytokinesis happen, so that the cell can pinch in the middle and become 2. Then we go all over again, but this time we don't have an interphase. We go straight to prophase 2. So in prophase 2, the two daughter cells line up again, and again we have metaphase. So this time no recombination occurs, no crossing over occurs. All right, so from here straight away they're going to go to metaphase, where they are going to line up at the equator. Mitotic spindle is going to come out and attach, and then in anaphase they're going to be pulled apart from one another, and then in telophase, essentially what happens is they're going to be pinched in the middle, and we're going to end up with 4 daughter cells. So in mitosis we end up with 2 daughter cells. In meiosis, we end up with 4 daughter cells. And each of these daughter cells, essentially, they are different to one another. So in mitosis, that's not the case because they're body cells, so it's going to be the same DNA. But in our gametes, in our sex cells, what happens is that the 4 granddaughter cells, they're going to be different to the parent offspring, they're going to be different to the parents, and they're also going to be different to each other. So all 4 are going to be different, and that leads to genetic variation. That is the key behind genetic variation. And that's this process. Alrighty. So again, if you have any questions, please don't hesitate to ask. Let's have a look now at cell replication. Let's begin by talking about the DNA structure. So essentially, remember, DNA is so based on the Watson and Crick model. We have so these are some key terms to remember. So firstly, we have nucleotide. So a nucleotide, one nucleotide refers to the phosphate, so it refers to the sugar there, to the phosphate, and then to the sugar there, the deoxyribose sugar, and the nitrogenous base. So remember, the biggest difference between DNA and RNA is that in DNA there is deoxyribose sugar, whereas in RNA it is ribose sugar. And also in DNA we have these particular bases, so ATGC, that is adenine, thymine, guanine, and cytosine. In RNA, what happens is that the thymine is swapped for a uracil, for a U. But that comes in a while, but let's just focus on the structures. There's the nucleotide. It is the alpha helix, so the DNA is wrapped really tightly. It's double-stranded. It is an alpha helix, and the complement and the base pairing that happens here, C pairs with G and A pairs with T. That's the only combination pattern that there is. C will only pair with G, so the cytosine will only ever pair with guanine, and adenine will only ever pair with thymine, and that leads to hydrogen bonding. So these bonds are pretty strong. So now let's think about DNA replication. What is actually happening? So remember. Sorry, I just remembered one thing, and that's one way to remember the pairings here, C and G, A and T, is to remember cars in a garage and apples in a tree. And that can help you remember the pairing there. But let's have a look now at DNA replication. So there are 3 processes to DNA replication. The first process is initiation. And initiation, of course, refers to the beginning of the process, and this is where our DNA is unzipped by the enzyme called helicase. DNA is wrapped really, really tightly. So it's wrapped really, really tightly, and obviously it's wrapped really, really tightly in the chromosome here, so that's what the chromosome is. It's basically the DNA wrapped up super, super tight, and it is protected by these proteins called histones, so it's really tight. And when we look at replication, we begin by opening up a certain area. So as you can see here, we begin by opening up this area, but remember, we're going to replicate the whole thing because we need the whole chromosome to be replicated properly. But here we go. When DNA replication begins, the enzyme helicase comes in and opens up the DNA. And then elongation is the process in which now we have primers that bind to the end of the DNA strand. And when they bind to the ends of the DNA strand, they basically urge this DNA polymerase right there to come and to start like bringing these strands, to start reading the strand and attaching the free floating nucleotides to each of the ones. So there's DNA polymerase. Here are the free nucleotides that are just floating around, and DNA polymerase is gonna read the strands. It's gonna go, okay, that's a G, so it can only pair with a C. That's a C, that can only pair with a G. That's a T, that can only pair with an A, and so on. And so that is happening. Now, a key part of the process here is that there is a leading strand and a lagging strand. So So DNA is wrapped in 5 prime, sorry, DNA is replicated in the 5 prime to 3 prime direction. So what that means is there's going to be a leading strand, which means it's going to be replicated moving forward, which is nice and easy. However, there is a lagging strand, which means this is where the replication begins. It's actually moving backwards. Now, as it moves backwards, the process is more, I want to say the process is like more disjointed because it's moving backwards, whereas moving forwards here is easier. So that's why we call it a lagging strand. And what happens is, because it's moving forward, sometimes it like there can be gaps in between, and we call each fragment that has been replicated like an Okazaki fragment, and that Okazaki fragment at the end is joined by another enzyme called DNA ligase. And so that is part of this whole process. Finally, we have termination. And in termination, essentially what happens is that the polymerase, right here, the DNA polymerase, is going to reach the end of the molecule, which symbolizes that the replication has occurred. It is going to then go away. The strands are going to recoil into a double helix. Remember, they like to be protected. They like to be sheltered, so they're going to recoil again. And then we're going to have a process called proofreading occur. So this is where nuclease enzymes are going to come in, and they're going to proofread what has been done to make sure that there are no errors in this process. And that is cell replication. Now let's have a look at DNA and polypeptide synthesis. And our inquiry question is, why is polypeptide synthesis important? So this diagram is the key here. This is our central dogma. So we essentially begin with DNA, where replication has occurred. We replicate our DNA. Once that has occurred, now DNA is important for our survival, and that's because one of the key things that DNA does is that it allows us to make the proteins that we need to survive. Most of the things in our most of the processes in our bodies require those proteins, like the enzymes that we have are proteins, the hormones that we have are proteins. So proteins are really, really important. So what happens is we have our DNA, and then we're going to use that DNA to have two processes occur. The first process is transcription, where we then change the DNA sequences from DNA to RNA. So this is where RNA comes in. And then once we have the RNA sequence, we then translate it to make proteins. So think of it like transcription is basically like writing down the recipe, for a specific dish that you want to make, and translation is then actually cooking that recipe in the kitchen. So let's have a look at what this really involves. So let's begin with transcription first. So in transcription, the RNA polymerase will bind to a promoter sequence. So whereas in DNA was the DNA polymerase, in transcription it's actually RNA polymerase. So here we are. So RNA polymerase right there will bind to a promoter section. Now here's the thing with polypeptide synthesis. It's the fact that we don't actually need to unwind the whole DNA to make our proteins. We only need, so at times we're only at different times we're going to need different parts. At different times we're going to need different proteins, which means we're going to open up different parts of the DNA at different times. So not the whole DNA is being opened in like replication, right? So in replication we replicate the whole thing. In polypeptide synthesis that's not the case. We are only taking a specific bit to produce a specific hormones. To produce a specific proteins. So let me just grab a quick sip. Okay, here we are. So once that has happened, our RNA polymerase will move along the DNA strand, and it will unwind a small region of the DNA. So this is the coding strand. So this is the strand that we actually want to code. So to get this strand, we're going to code its complementary strand so that we can get this combination here. So RNA polymerase is going to read that DNA strand that we had, the DNA template strand, and then it's going to match the complementary free-floating nucleotides to create an mRNA chain. And it does that. It creates the mRNA chain. Now remember, two differences between RNA and DNA is that in RNA there is ribose sugar in comparison to deoxyribose in DNA, and RNA is also going to have uracil instead of instead of thymine. So uracil replaces thymine, and that's sort of the key there as well. Okay, so what happens is that once that has happened, once the RNA polymerase reads the DNA template strand, it matches the complementary free-floating nucleotides to create an mRNA chain. And mRNA stands for messenger RNA, so it's basically taking a message. Think of it that way, it's a messenger. And then once the process is done, the terminator sequence ends the transcription of the DNA. So there is a terminator sequence, and as the name suggests, it stops the process because we don't want, again, we're not unwinding the whole DNA, we are only taking a bit of the protein a bit of the sequence that we need to make our protein. And finally, once that is done, we go through a process called post-translational modifications. So what that means is we are going to splice out the bits that we don't need. So the DNA, the whole DNA does not code for proteins. It does not code for proteins because it has other like junk sequences as well. So what we do is once we have our strand here, we realize, for example, we may not need some of these introns, some of these things here that don't actually code for protein. So we can splice it out, we can cut it out, and that way we only have the DNA that we need. Sorry, we only have the RNA that we need because it's in RNA form. It's in mRNA, so it's a mature mRNA. So mature mRNA is when it has gone through its post-translational modifications. Okay, so that brings us to translation. mRNA is the scribe, right? It is the messenger. It has written down the recipe. Now it's going to take that recipe to the ribosomes, to the kitchen. So once you write down the recipe, you found a recipe on YouTube that you really like, you wrote it down, and now you're going to actually go ahead and cook it. But to cook it, you can't just cook it in your living room, you can't cook it in your bedroom, you can't cook it in your balcony, right? You actually need to properly cook. You need to go to the kitchen where all the equipment is. And that, in this case, is the ribosome for the body. So, for the cells, rather. So, it's in those ribosomes where we actually see the last part of this process play out. That's where translation happens. So, what the ribosome does is, here we are. So this is in the ribosome. This is our messenger RNA. Messenger RNA has come to the ribosome. It's like, hey, hey, here is the sequence for the proteins we need to make. Ribosome's like, all right, I'm on it. What that does is that the ribosome matches complementary tRNA molecules to mRNA. As soon as it says I'm on it, we see this molecule tRNA come in as well. And basically, tRNA, what we have here are these anticodons. So the ones on the mRNA are known as codons. The ones on the tRNA are anticodons. So the anticodons and the codons are going to match to one another. And with each anticodon, we're going to produce a specific amino acid based on the sequence of the anticodon. So as each subsequent RNA docks to the mRNA here, it's going to lead to a polypeptide bond. So it's brought an amino acid, it's going to dock. That other one's brought an amino acid and it's going to dock. And these amino acids are going to form bonds between them, and that's what we call a polypeptide bond. And a polypeptide chain is elongated by continued addition of amino acids. So that keeps happening. We keep finding these finding these amino acids, right? And it keeps sort of lining up. Now remember, amino acids are the functional units of proteins. So that's what amino acids are. So essentially, once that chain is elongated, we are going to reach a stop sequence. Whenever that protein is finished, we're going to reach a stop sequence, a sequence that tells it to stop this process so that it can be released. Once that happens, the ribosome releases the RNA. So once that stop sequence is reached, the ribosome here is going to let that mRNA go, and it is going to also release this polypeptide molecule over here. And as a result, then, this polypeptide molecule is going to fold over in different shapes. It's also going to undergo post-translational modifications, and then that will result in a mature protein being ready to use in the cell. So protein structure and function. So the fundamental unit of a protein is amino acids. They're the functional units, and they have various side chains. The primary structure is when there is a sequence of amino acids in a polypeptide chain. The secondary structure is the formation of alpha helices and beta sheets. Tertiary structure is the formation of overall 3D structures and side group interactions between these amino acids for maximum stability. And finally, the quaternary structure is when multiple protein subunits are involved. They are interacting. So what are the functions? Protein is important for spore and structure. So in terms of macromolecular substances, keratin is protein, and so is tubulin, which is micromolecular. It's part of the cell. Enzymes are also made of protein, so they are biological catalysts and we need them. And a really big example is ATP synthesis, which allows us to produce ATP, which is energy. Antibodies are an essential part of the immune system, and again, they are proteins. They're made of proteins, and that includes things like immunoglobulins. We have messengers, so that includes our hormonal messengers like oxytocin. And finally, protein is really important for storage and transport. So moving molecules into cells and around cells requires the production of these stores. And an example of that is ferritin, which stores iron. Okay, genetic variation, let's have a look. So with genetic variation, you would have looked at autosomal versus sex-linked inheritance. Autosomal inheritance is when a gene is carried on the 22 chromosomes there. So in the human body, there are 46 chromosomes. And when, for example, when you talked about fertilization, when an egg meets the sperm, there are only 23 chromosomes in a sperm and 23 in an egg. Those, when they fertilize, when fertilization occurs, it essentially leads to an organism which is going to have 46 chromosomes, which is the number that we have. And so this is what the chromosomes essentially look like. So autosomal chromosomes are the first 22. Alrighty, that's the first 22 chromosomes right there, and they are going to carry genes that will not be passed on to the next generation because they're autosomal. So somal, remember, somatic, it's only going to affect the somatic cells. Sex-linked chromosomes, though, this is where the genes are carried on those little 2 babies, not 22, on the 2 right there. So women have an XX chromosome, males have XY chromosome. So here we are. Now, based on that, we can then talk about the expression of these of phenotypes. So, for example, a dominant trait is one that will always be expressed over other traits. That's what we call a dominant trait. Recessive trait is when it requires 2 of the same alleles to be inherited in order to be expressed. So 2 of the same alleles have to be inherited, and therefore it's recessive. Codominant inheritance is when both alleles in a gene pair are fully expressed. So both of them are expressed. So imagine, for example, a white and brown cow. In terms of codominance, an example would be a cow which has both white and brown spots. Incomplete dominance is when one allele isn't expressed over the other, and a good example of this is, for example, when we mate red and white flowers together, we can end up with pink flowers because it's incomplete dominance by both. It's not white, or nor is it red, nor is it white and red. It's a mixture of the two, and so that's pink. Finally, multiple alleles. Multiple alleles is when one allele is not completely expressed over its paired allele. One thing I will again quickly clarify when we talk about alleles is that, think of it this way. An allele is basically like a flavor of certain things, right? So for example, we have a gene for eye color. The alleles can be blue or brown or green or hazel or whatever. So the way I like to think about it is like Coke flavors, right? So Coca-Cola flavors. So for example, if we've got, so basically if the gene is Coca-Cola, the alleles are going to be like 0. It's going to be the original Coca-Cola, that's another allele. Or it's going to be Diet Coke, that's another allele. So it's basically like the different variations of it. That's what an allele is. So when we come across questions that ask us to work through these inheritance patterns and so on, we make use of pedigrees. Pedigrees help us trace phenotypes through generations, and when we come to draw these, we always want to include a key, and we also want to label generations. So this is what it looks like. So essentially, as you can see here, we have a pedigree. There is the marriage line between a female and a male, and they have 2 descendants. They have a son and a daughter, and that's a sibling line between the two. So this is how we draw pedigrees based on the information we're given, and it helps us determine inheritance as well. Here are some key tips when it comes to actually deconstructing pedigrees and answering questions about pedigrees. Firstly, remember that recessive traits are going to skip a generation. So recessive traits can skip a generation. Dominant traits cannot skip a generation at all. Like, it's not possible for dominant traits to skip a generation, and that is because if it's a dominant trait, if it's a dominant trait, what happens is if it's homozygous dominant, then we know that the trait is going to be a phenotype. But even if we have heterozygous, we know it's the dominant is going to win because the dominant will be expressed. So remember, when we actually write this down, and I might just get my pen out. Here we go. Alright, when we actually do this, remember, when it comes to this, when we talk about the traits, when we talk about a dominant trait, we refer to it with a capital. When we talk about a recessive trait, we refer to it with a lowercase letter. So for example, if it's a recessive trait, we will say it's HH. Please pardon my H right there. There we go. HH, yep. So there are 2 H's right there. And it is recessive. Okay, they're both. So we just basically sort of to showcase that they're both going to be weaker than the capital one here. If it is dominant, it is going to be, it can either be, now in the case of dominance, where is my cursor? Okay, in case of dominance, it could be either H or big H and please. Okay, that's pretty terrible. Give me 1 sec to take that off. Here we go. Okay, or it can be both capital letters right there. So there you are. So remember, this will be homozygous dominant because it's homozygous, so they're both the same alleles. They're both dominant. And this will be homozygous recessive because, again, homozygous, homo, they're both the same, and they're both recessive. This is going to be heterozygous because we've got one dominant allele and one recessive allele. But even if you have one dominant and one recessive allele, think to yourself, which one will win when it comes to expression, right? When it comes to phenotypic expression. And the answer here will be the fact that this dominant allele is going to win. And best way to get better at this is to practice. These can be a bit tricky, especially this and Punnett squares together can be a bit tricky. So practice lots of this so that you feel confident and comfortable with them. So wonderful. Now, Punnett squares, essentially they allow us to theorize how genes may have passed. And often in exam questions, you might find questions where you are given like a pedigree, but in order to determine the inheritance patterns in that pedigree, you're actually going to have to draw Punnett squares first. So that's where we want to keep that in mind. And yeah, so Punnett squares allow us to generate information about potential genotypes and phenotypes, and we always use a checklist when it comes to these. And finally, inheritance patterns in a population. So let's keep going so we can get to module 6. This is where we think and we consider how can a population's genetic patterns be predicted with any accuracy. So this is where we have two techniques we need to know when it comes to inheritance patterns. The first one is DNA sequencing. And how does that occur? With DNA sequencing, we begin by isolating DNA from cells. We get its sequential order of the nucleotides because that's what makes a DNA unique. It's those nucleotide pairings. And then we put in a computer, we process it, and we can then compare whole genomes of individuals. And we can also conduct transcription and translation of genes in silicio. And so, as a result of this, we get a lot of information. We get information surrounding single nucleotides and single nucleotides and their polymorphism. So if there has been changes in single nucleotides, we also get information about understanding that each individual has a different genome, and we can use that to then identify differences and similarities. Modern computation will also allow for patterns to be processed, and with those patterns we can then trace inheritance of genes, alleles, and also SNPs, so single nucleotide polymorphisms. So the change in a single nucleotide, which is then going to lead perhaps to a bigger change in the whole sequence. Wonderful. And then we've got DNA profiling. So DNA profiling, probably you might be familiar with it in one capacity or another. Definitely if you watch crime shows and thrillers, that's where you would have seen DNA profiling a lot. So this is where we collect samples from individuals. So common practice, for example, includes collecting blood, hair follicles, or mouth swabs. We digest that DNA. So basically what we're doing is, once we get that, we're going to cut up the DNA into small pieces using what we call a restriction enzyme, and that restriction enzyme will cut DNA in specific places where we want it to cut, and then once that happens, we are going to put it in this machine called gel electrophoresis. So in this, I'm still on my pen cursor, there we go. Let me go back. Okay, oh nice, I've got a laser pointer. Here we go. So we then have this machine called gel electrophoresis, right? And so in this process, gel electrophoresis, what we do is we put DNA here at the sort of the front of the machine here, and it goes from positive to negative. What happens is that we put in the DNA, we've got these samples, and we've got a marker that we use, and that DNA will then travel according to its like what its like constitution is. Usually we see that smaller pieces of DNA travel further in comparison to bigger parts of it. So anyway, so it travels from positive to negative, and then we're going to see travel. And using this, we can then compare this, for example, for fingerprints, and we can do that to compare fingerprints to sort of determine, you know, like to determine crime scene related suspects, to determine paternity. That's also where DNA profiling is used, to compare paternity and to determine if you know two people are related or not. So if you have a sample here, it's a crime scene, we have a sample of a hair follicle that was found on the um, on the on the crime scene, right? And we've got 3 suspects, we got their hair follicles, and now we have put it through, and essentially we can see that we want to see which ones match up to this initial, to the marker here, to the hair follicle that was found in the crime scene. And you can see that it's actually C that matches the most to the marker here. So whoever is C here in this scenario is the person who has who is responsible for that crime. Now, one thing to be mindful of with DNA profiling is that historically it has known to have lead to like to be complacent, in the sense that there have been wrongful accusations, but for the for the sake of the module, for the purpose of the module, we just have to know what the process is, and we just have to focus on what it is and how it occurs. So the degree of similarity equates to the degree of relatedness, and using this we can observe conserved regions of the DNA. So the conserved regions are the regions that are actually different between everyone. So we actually share a lot of the DNA, like a lot of our DNA is very similar, but we have conserved regions that then lead to our uniqueness. And it's also effective in humans because we have large stretches of junk DNA that we can use for this process. So remember I said earlier, not all DNA makes protein, and so we have junk DNA as well, which we can use for this sort of process. Now let's have a look at inheritance patterns. So this is what you're going to do. You're going to look at large-scale collaborative projects. You're going to look at how they have identified trends, patterns, and relationships, and how they have used that and applied that to have outcomes. So essentially, there are 3 processes that we're going to look at. Firstly, we have conservation management. So conservation genetics is the field of bio where essentially we combine molecular genetics, ecology, and biodiversity to propose species management. And it is used to, this process uses population genetics to identify any alleles that are at risk and proposes ways to preserve the biodiversity. And an example of a group that does that is the Australasian Wildlife Genomics Group, which is found in the Sydney University, and they have projects like the Devil Tools and Tech projects, the Koala Genome Project, and also the Marsupial Genomics. Okay, next up we have inheritance of disease. So we can actually utilize this data to look at the inheritance of diseases. So what we know is that statistically there is only, and it's crazy, there is only a 0.8% of nucleotide variance among humans. So only 0.8% of a DNA is actually different to one another. And like I said, that's crazy. That means a lot of it is very, very similar. So what we know is that if diseases are arising, it actually would be because if diseases are arising and not everyone has the disease, but some people do, that means that lies in the 0.8% of variance, because it wouldn't be in the 99.2% which everyone has. It's going to be in the 0.8%. So we then utilize that idea to understand the cause of genetic diseases. Why is it? Is it due to random mutations that occur, or is it due to the interaction of alleles? And then we can use that to design therapies to repair genetic disorders. And a good example of this is the Human Genome Project. So the Human Genome Project collated data on thousands of genomes to identify genes, variable regions, haplotypes, and they have identified genetic causes of breast cancer, Alzheimer's, and also colon cancer. Alrighty, looking at human evolution. So Finally, we know that cultural and regional groups are linked by the prevalence of particular haplotypes. So essentially that we're linked by these particular groups of similar genetic sequence, and we can map that to trace the movement and evolution of human species. And we've actually been able to do that to determine the origins of mitochondrial Eve, who we said that lived in Africa 20,000 years ago. And that is part of the International HapMap project, which uses human genome data to identify SNPs, haplotypes, and haplogroups, so groups essentially of similar genetic constitutions. And so from this information we can trace back lineage. So, top 5 tips for Module 5. Know your processes. Those processes of meiosis, mitosis, DNA replication, polypeptide synthesis, they're all super important. Also know your basic skills. So, how to draw Punnett squares and pedigrees. Again, really important. Always think about the broader context of genetic variation. So, how do we achieve variation every time and at every stage of like cell division, gene expression? And memorize those examples. Memorize the methods of replication, functional proteins, the methods of inheritance, the technologies like the DNA sequencing and DNA profiling, and then be also memorize examples of how we've used this information in inheritance patterns, and link that to large-scale collaborative projects that we have essentially found. Alrighty, in terms of now applying the content, do practice questions. So there are lots of practice questions that are available for Module 5 in the past papers. You just have to figure out which ones link back to the syllabus. If you just type up, because remember, the biology syllabus had a syllabus change in 2019. And so what that means is that we need to make sure that, so that means that you only have, 4 years now worth of papers, counting last year's paper, which still, better than, for example, what the class of 2019, 2020, 2021 had. However, you can find, it's still pretty limited, only 4 papers. So you can find lots of past paper questions in past exam papers before 2019, but just make sure that they link back to the syllabus and you're not like studying for anything that won't be tested, because it is part of the older syllabus, those older papers, but you'll find Module 5 stuff that you can use. Alright, and we of course have lots of questions available in our topic test books and also in our NEEP exam papers that are available on 8notes Plus. So now let's have a look at Module 6. Finally, we are here. Module 5 just usually takes a lot of time because it's so dense and there's so much to go through. But I'm hoping you guys feel refreshed with all that Module 5 review. Please ask if you have any questions. So let's begin by looking at Module 6. So we're looking at 3 specific dot points. We're looking at mutations, we're looking at biotech, and we're going to look at genetic technologies. So we are going to focus on these specific questions. We're going to look at how does mutation occurs. We are looking at what are the possible outcomes of a mutation. How can we actually manipulate the knowledge of genetic change for our advantage? We're then going to look at what is the deal with the field of biotech. What exactly is biotech? We're going to look at what are the cool genetic technologies that we have developed and their uses. And then in the last part of genetic technologies, we look at the social, economic, and environmental and cultural implications of genetic technologies. So let's get into it. Let me just grab a quick sip. So our inquiry question is, how does mutation introduce new alleles into the population? So Module 6 is a really nice segue from Module 5, where now when we look at mutations, firstly we're going back to our processes of replication, translation, transcription, and talking about mutations affecting those processes. Okay, here we are. So first question we have here is that we're going to look at is, how do mutagens operate? the different types of mutations. We're going to talk about somatic versus germline mutation, and then talk about the effects of mutations on coding versus non-coding DNA, and then look at causes of genetic variation, and finish off by talking about population genetics. So we're going to talk about mutations, gene flow, and genetic drift. So here we are. Our inquiry question is, how does mutation introduce new alleles into a population? Remember, a mutation, firstly, is a permanent alteration to the nucleotide sequence of an organism's genome. This is where we are permanently altering someone's, or rather, natural circumstances or their own system leads to the alteration of someone's own nucleotide sequences, which then changes the proteins that they are able to make. So we'll firstly begin by looking at the mutagens. What are the things that actually cause these mutations to occur? And we're going to talk about electromagnetic radiation first of all. Electromagnetic radiation, as the name suggests, refers to the waves that are emitted by electrically charged particles. They interact and ionize atoms that they encounter, and there is a danger, and the danger of radiation lies in its penetrative ability. So electromagnetic radiations can penetrate quite deeply, and they can travel into the nucleus, and they can adhere with DNA molecules, and they can lead to the creation of free radicals. So it's not very good at all. And how does it actually cause mutation? So once it actually penetrates in, there's a lot of energy, right? Electromagnetic radiation, there's a lot of energy input, which actually may alter the way that the DNA is bonded, and it can change the chemical composition of the DNA as well. It can also lead to the rearrangement of the structure. So here we are. There are examples. For example, ultraviolet light is an example of electromagnetic radiation, and it is radiation from the sun which, if encountered in high doses, in daily high doses, can be harmful. However, we do encounter it daily, and that's why we know that there is huge stress on the importance of wearing sunscreen, being sun safe, wearing protective clothing, because we know that there is an increased risk of melanoma from it, which is skin cancer. So UV light is harmful in high doses, and what it does is that the incoming photons actually excite the adjacent pyrimidines. So these adjacent pyrimidines, which includes T and C, so thymine and cytosine, so it incites them, and it then causes them to actually turn around and bond to each other through covalent bonds. So instead of the hydrogen bonding here, that hydrogen bonding is broken so that the C and the T can bond to one another. And on an average summer's day in Sydney, a person will acquire around 20,000 pyrimidine dimers. So it does happen. It is a very, very common thing. But, and there are lots of mutations that occur, but the thing here is that we do have our own repair systems that do repair some of these changes. So we'll talk about what happens if those changes aren't repaired. But this is the first mutagen that we look at, electromagnetic radiation. Now let's have a look at chemicals. Chemicals are also mutagens. We have radioactive agents, so agents like uranium, and they can release radiation, including alpha and beta particles, and they are going to be high energy particles, and they are going to they can include alpha and beta particles, which are high energy, and they can also include gamma waves, which is actually a form of electromagnetic radiation. And both of them can then, or all three of them can then penetrate the cell and interact with the DNA bonds, again creating structural disruptions. In terms of metals, so the metals that mutagens include arsenic, nickel, and cobalt, and so being exposed to them for a long amount of time, it can affect the integrity of the DNA repair system. So we have our own inherent repair system that essentially stops the proteins from that basically fixes any mutations or tries to fix as many mutations as possible. But if that repair system is affected, which is the case with metals, that they can affect the DNA repair system, they can actually stop proteins from being able to recognize base pair mismatching. So it stops that recognition of base pair matching. So any incorrect base pair matching, it stops that from being detected. And so as a result, we have as a result, that's overlooked and that mutation goes unnoticed. And it reduces the fidelity of the DNA replication, so more errors occur. And nickel especially inhibits the ability for histones to condense DNA. So histones are really important because that's where they're wrapped around DNA and they essentially protect our DNA. And if they essentially, and if nickel affects the ability for histones to condense that DNA to get that DNA to be protected, it can then lead to the chromosomes being in a bad formation, and that can of course affect the protection of the chromosomes. Last one, naturally occurring mutagens. So naturally occurring mutagens includes viruses. So these are things that are out there in nature that can lead to mutations. So viruses are an example of this, and what viruses do is that viruses replicate by inserting their own genetic material into the host cell. So they insert their own genetic material into the host cell and they disrupt the normal cell function. And by doing that, that can lead to lasting changes to an organism's genome. So viruses don't have the machinery to actually make their own proteins. So every time a virus, so they need basically another body to survive. Like they need a human body to survive. So they get in there, they're going to get into our cells, gain entry, hijack our whole transcription translation machinery to then put in their own genetic material so that that material can be produced, so that their proteins can be produced. And these are some examples of viruses. So HIV, of course, which then leads to AIDS, hepatitis B, Ebola virus is another one, adenovirus, influenza, and bacteriophage. And bacterial infections, which is bacteriophage right there, what they do is that they induce inflammation. So they cause inflammation, and that reduces the efficiency of our DNA repair systems. And it also then leads to the increase in mutations. And a good example of this is, for example, Helicobacter pylori, which is a bacteria, and it has actually been linked to the development of stomach cancer. Remember, cancer is that unhindered replication of the cells, right? And what happens is, if our DNA system is repair system is unable to repair that mutation, the cells keep replicating, and then that then leads to cancer. And in this case, there has been correlation between Helicobacter pylori and the development of stomach cancer. So now we know what these mutagens are. Let's talk about the effects of these mutagens. What exactly happens? So sorry, let me grab a quick sip. Alrighty. So here we are. We're going to now compare the causes, processes, and effects of different types of mutations. But we're first going to talk about point mutations. So point mutations, in terms of what causes them, firstly, they are caused by external mutagens, DNA replication errors, and DNA repair system malfunction. So point mutations are these mutations in the coding sequence. So they just affect one or two nucleotides. It just affects the coding sequence here, and it can have these specific effects. It can lead to a silent mutation, which means we don't actually see it having an effect on the amino acid sequence that's produced. It can be missense, nonsense. It can lead to the creation of SNPs, which is a single nucleotide polymorphisms, which is the change in a single nucleotide. It can also lead to the creation of new alleles. So mutations are not always bad. They are important for variation, but they can be bad. They can be potentially beneficial, and they can be potentially negative. And just because a point mutation is small doesn't mean that its effects will be small. So cystic fibrosis is an example of that. So here we have point mutation, an example of what actually happens. This is the mechanism, and this is the effect right there. So essentially what happens is that in point mutations, this is a normal codon sequence, right? So we've got, let me use a highlighter. Okay, so we've got AGC, we've got AAG, and GCU. We've got 3 specific sequences right there, and it produces these proteins, right? So these specific amino acids, which will then join together, form polypeptide bonds, lead to a polypeptide change, form proteins. So there are 2 types of point mutations that can happen. There can be substitution, or there can be a frame shift mutation. In substitution mutations, what happens is, as you can tell by the name itself, one of the nucleotides substitutes the other. So instead of the G here, the guanine, there has been a substitution, and there's been a uracil that has been inserted. And this, remember, this is happening during translation. So because of that uracil insert, or the transcription has occurred. Translation is happening now, but during transcription there's been that change of uracil. From guanine we've gone to uracil, and because of that the code now changes to the codon changes to the anticodon, the codon changes to the anticodon, my bad. The anticodon changes to AUC instead of AGC, and that means that our amino acid would also change right there. Frame shift mutation can either be insertion or deletion. So frame shift means that it just shifts one over. So it's causing a downstream shift, basically, in reading the sequences. So how does that happen? They can either be insertion or deletion. In terms of insertion, what happens is that we have the ACG. So here we have inserted a C just before the G there, and that has, instead of changing just one amino acid, it's actually changed the whole sequence here. Same thing happens if a deletion happens. So we've deleted the G here, and that has also changed the whole sequence there. And its effects can be the following. This can be the effects of it, right? It can either be missense, which means that in this case here, the mutation edits the mutation which edits codon sequence to incorporate different amino acids in the protein. So there is a different amino acid that has been added in, and that is so that's an example of missense. This is an example of missense, and this is an example of missense. So a different amino acid has been incorporated in. Nonsense mutations, like the nonsense effect, essentially is when a premature stop codon is introduced. So when you look at this, you will actually look at a table that tells you, there's a table that actually tells you what the combinations of these anticodons, what amino acids that they'll lead to. And if you want to check it out, you can just type up amino acid translation table or something. It should pop up, and you could see, for example, that the codon here, AUAG, that is a stop codon. That is telling us that, okay, the process stops. And what it's done is, as you can see, it has prematurely stopped the process. It has prematurely stopped it, and it has prematurely terminated the polypeptide synthesis. A silent mutation, on the other hand, is when, luckily, yes, there has been a change here, but if you look at the amino acid table, you'll see that there can be a variation of sequences, of anticodon sequences, that lead to the production of one amino acid. So AGU also leads to the amino acid SER, that I don't know the full form of SER, but it leads to the production of this specific amino acid, and so does AGC. So it's like so there are a couple of anticodon combinations that will lead to the same amino acid being formed, and so as a result, luckily, there is no effect on the amino acid sequence. Okay, now that was point mutation. That's when when the mutation affects the nucleotide sequences. Point mutations is the first one we looked at. The second thing we're going to look at is chromosomal mutations. This is where the chromosome, the entire chromosome, is affected, and there are essentially two main causes for it. Generally, it generally is a result of errors that occur in meiosis, in the process where the gametes are being formed. So essentially, firstly, there could be an issue with crossing over, and if crossing over occurs incorrectly, that means that chunks of DNA can be screwed up, like they can move places, and that can lead to structural mutations, these mutations right here. Secondly, there can also be issues with sister chromatids being incorrectly separated. So remember, when we've got the sister chromatids that line up near the mitotic spindle, that line up and the mitotic spindle is attached to them, they're separated. If there's an issue with separation, or they may not be separated, or they may just be separated for like one side, that can lead to an uneven number of chromosomes, and that leads to mutation in the number of chromosomes. The effects of this are usually severe, more severe than point mutation, because this is where the whole chromosome is affected, so it can affect a number of genes. So these are the structural mutations that can occur. So when the chromosomes go to cross over, for example, it could mean that there's a deletion of a chunk of the chromosome. Inversion, they can like invert over here, as you can see. Translocation, a part of the chromosome here can translocate to the other chromosome, and duplication, it can duplicate as well. So it leads to so that's structural mutations. The second one is mutation in chromosome numbers. So for that, there's either aneuploidy or polyploidy. So aneuploidy is when the overall chromosome number of the offspring is different to the parent organism. So in the case of Down syndrome, the individual has 47 chromosomes instead of 46, because they have 3 chromosomes instead of they have 3 copies instead of 2 for trisomy 21, which is chromosome 21, so they have 3 copies of it instead of 2. In terms of polyploidy, what happens is that when an organism contains more than 2 sets of homologous chromosomes, so instead of having 2 chromosomes, they actually could have, for example, 3 copies of every homologous chromosome instead of the 2. And an example of that is triploidy, the condition. An example of aneuploidy is Down syndrome. Okay, awesome. And now let us talk about somatic versus germline mutations. You kind of would already be familiar with this specific concept from module 5. So germline mutations, it's going to affect the germ cells, the gametes, right? So for example, these are the parental gametes. They're going to fertilize. There's a condition, let's say here, that there's a trait specifically that's carried in the mother's in the chromosomes here, in the egg, and so when fertilization occurs, there's an embryo, the entire organism is going to carry that mutation. And because the entire organism carries that mutation, when that organism reproduces, half of their gametes will carry that mutation, which means that depending on whether it's dominant or a recessive allele, the offsprings may have that mutation, because it's affecting the germ cells, it's affecting their sex cells. On the other hand, somatic mutations is a somatic mutation. Somatic mutation is a somatic mutation. No, somatic mutations will only affect body cells. So what that means is, here if you have a mutation in some cells over here, it's only going to affect this area. It's not going to affect the offspring of the individual, because the mutation is in their somatic cells, it's in their body cells. The mutation isn't in their germ cells, it's not in their gametes, so then it won't pass on to their offspring. So there we are. All I just said. So germline mutations affects the gametes, and so changes to the gametes leads to changes, changes to all cells of the offspring, and the offspring will inherit mutated information, and it can be caused by both external and internal factors. An example of that is cystic fibrosis. Somatic cell mutations are when mutations affect your somatic cells, your already existing cells, your body cells, and so that means that it's not going to affect the future generation. So example of that is skin cancer, where for example, if an individual has skin cancer, it won't be passed on to their offspring, because it only affects that individual's somatic cells. But there could be like genetic factors and stuff that means that the individual may be more susceptible to it, but that's different. Like they wouldn't have it just because the parent had it. Whereas in germline mutation, the chances of the offspring having the condition are pretty high, because depending on whether it's dominant or recessive, because it's passed on through the generation. Okay, so now we're going to look at, and here we go. Okay, now we're going to look at the significance of coding versus non-coding DNA sections of the DNA. So remember, not all of the DNA is coded for by proteins. Not all of the DNA codes for proteins, my bad. So what we have here is that coding DNA, in fact, is very small. Like it's a very small part of the overall DNA. Like a very small part of DNA codes for genes. Very, very small. That's our coding DNA. The rest of it, we have junk DNA, we have enhancers and silencers, we have promoter sequences. That name might stand out because remember we talked about how in DNA replication and in transcription and translation, we have promoter sequences that actually tell us when to or where to actually start these processes. We have introns, terminator sequences, and non-coding RNA as well. So here is the purpose of each. So genes are, remember, these mutations that occur to our genes, to a coding region, it can have an effect on the protein that is produced, and it can have a serious effect on cell function. Mutations that occur to the introns, they may have effect on splicing of genes. Remember how when we talked about post-translational splicing, we talked about how we take out the introns so that we are left with the extrons, coding DNA, so it can have an effect on the splicing of the gene. Promoters, mutations to promoter region can actually inhibit the ability for a gene to be transcribed, because remember, those promoters are telling us where to actually start the process, where to actually start transcribing. So if there's an issue here, that will have an effect. Oops. Oh, my bad. Sorry. Let me just quickly forward. Oh, that was fast. Perfect, and my highlights are still there. Awesome. Alrighty, even though they're terribly done, sorry about that, but they're still there. So, we talked about promoters. Now again, terminators, same thing as promoters. Mutation to a terminator sequence can result in incorrect or elongated mRNA sequences, because it doesn't know where to stop, so it can be incorrect and elongated. Mutations to enhancers and silencers, so these are the regulator regions. They actually they enhance certain genes and they silence certain genes when we don't need them. So if mutations occur in this region, it can actually lead to some genes being over or underexpressed, and that can lead to either too much protein being formed or too little protein being formed. Non-coding RNA is when mutations occur to non-coding RNA sequences, and that can result in alteration to important molecules. So non-coding RNA is important for those RNA molecules that we talked about in the processes of replication, transcription, and translations, like tRNA is an important one, right? So if there's a mutation to that, it can affect the function of like tRNA and mRNA. Finally, junk DNA. Mutations to junk DNA, as the name suggests, won't have a huge effect, and they probably won't matter. So we can see how mutations, for example, I'll change color here, mutations to obviously genes, to introns, to promoters, to enhancers and silencers, to terminators, and also to non-coding RNA, pretty much to most of them, and almost all of them, mutations will have an effect. And that tells us that it's not the best, right? Like, it's not ideal to have those mutations. However, we will talk about how actually mutations, at the end of the day, they lead to genetic variation. And this is one of the key concepts here, is that the aim is for genetic variation for the continuity of species in a way. Right? And I'm kind of linking this back to module 5 here, but that's the key there. So we want to now talk about how through these different processes genetic variation is achieved. So mutation leads to the creation of new alleles through point and chromosomal mutations. Even if those alleles may not be the most beneficial, it still leads to the production of those alleles. Meiosis is when those alleles are recombined and sorted in random independent ways. So remember, we have crossing over, we have random segregation. So the way in which those chromosomes are separated is random, and also the way in which they're sorted and separated is also random. So that's independent assortment and random segregation. Crossing over, really important. You know these three processes in detail. Like, I would add that to my notes. And finally, fertilization. So this is where these variable alleles from two different organisms are brought together, and they interact in new ways to lead to the creation of new alleles and new phenotypes. So there is an uncontrollable combination of a number of possible gametes, and the interaction of alleles to produce phenotype also leads to genetic variation. Awesome. Now let's have a look, and I know it's a long dot point, but I believe this is one of the last parts of this dot point. So let's have a look at the effects of mutations, gene flow, and genetic drift on the gene pool of populations. So here we are. So a mutation, remember, is a permanent alteration to the nucleotide sequence of an organism's genome. It leads to the creation of new alleles. It can be favorable, and it can also, it may be favorable and subsequently selected. Gene flow is the transfer of genetic variation from one population to another, leads to changes in the frequency of alleles, and it can affect the distribution of genetic diversity. And finally, genetic drift is when the frequency of alleles within a population changes because of either disappearance of particular genes, and that could be a result of natural selection over time. So, for example, traits can become more or less prominent, and it can also lead to evolution of species and creation of new species as well. So when we talk about genetic drift, let's just talk about a couple of causes. So that, and this is kind of like a bit of revision from year 11. So bottlenecking is a process in which there is a abrupt reduction in the number of individuals. It could be due to natural causes, a sudden severe selection pressure, and that leads to a loss of diversity. The founder effect, of course, is when a new population is established by a small number of individuals. So they separate from the main population, they establish a smaller population, and it leads to a loss of genetic variation within the new group, and it can also lead to new speciation events as well. Evolution, overall, remember, is the change in a species over many generations. It results, and as a result of natural selection of favorable characteristics. And remember, when we link this back to evolution, it's because we're talking about mutations, how it leads to genetic diversity, and how it can then lead to these specific processes. Remember, variation occurs within a population due to natural selection. My bad, right there, natural selection. Environmental pressure is applied to population, and phenotypes best suited to the environment and the changed environment will survive, and surviving organisms will reproduce. So this is telling us that genetic variation is super important, and I'm trying to draw a straight line, but that's not working. So genetic variation, GV, really, really important. And I want you to remember this because we're going to come back and link this back to biotechnologies and genetic technologies when we discuss that. Here is a practice question from the NESA's sample paper. We won't go through it because we're a bit tight on time, but what I will do is I'll leave that there, and of course remember you have access to the slides, and yeah, you have access to the slides to have a go at it. And this is from, like I said, this is from the sample NESA paper, so I think you'd also find a little explanation for each of these points as well in that sample paper. So if you just type up biology additional questions, sorry, yeah, not sample paper, my mistake, NESA additional examination questions, biology HSC or something thereabouts, you'll get access to this. It's like an 80-page long document, and there are a range of questions on there. I might put it in the chat when we get up to this point in the recording, so feel free to remind me if I haven't already. So yeah, I'll put it in the chat, but you can access that, and it's got a little explanation as well. What I will do though is I'm gonna move on. Actually, let's do this part together. So here we have a practice question. So compare the processes and effects of point mutations and chromosomal mutations. Include examples in your answer. So sample answer can we can talk about essentially begin by talking about the fact, like, what mutations are, right? Define what mutations are and how they occur. So here we go. All mutations are changes to DNA. They occur during DNA replication, during mitosis, and during meiosis. So we actually identify what these processes are. In biology, always identify and give definitions for the terms in your response before you go ahead. Definitions are really important. We then have our first couple of paragraphs on point mutations. So as you can see here, it's not just one big chunk of paragraph, and that's because usually when you write a big paragraph, in cases like this for like an 8 marker, it can start to lose meaning, it can start to lose structure, so always recommend doing like smaller paragraphs. So here we are. Point mutations are changes that occur in a single nucleotide. These changes can be substitution with the wrong nucleotide, an extra nucleotide being added, so that's addition, or insertion, or a nucleotide not being included, which is deletion. So we've talked about 3 types here. So we've talked about point mutations and how that's either substitution or frame shift, which includes insertion and deletion. It is possible to have multiple point mutations along a chromosome. The order of nucleotide bases determines the protein that is produced by the cell. So we actually link this back to the effect. We have to actually talk about what is actually happening here. The point mutation may have no effect on the protein being produced, as the change may still enable a triplet code for the same amino acid, or change of one amino acid might not have a significant effect on the resulting protein. So here we are. That is, can you identify which type of effect they're talking about here? It may have no effect. And so they're talking about the silent effect here. So that's silent S. The point mutation may mean that the triplet code initiates a stop sequence, in which case the protein will not be produced. It may mean that a range of proteins are not produced at all, or the greater quantities of proteins are produced, and that will be nonsense mutation. And finally, frameshift point mutation is caused by an addition of deletion, is caused by an addition or deletion. Every triplet on the DNA after the point mutation is affected, this can radically change the protein product of the cell. And this is just a quick little summary on point mutations. And then we talk about chromosomal mutations. And remember, it's asking us to compare, right? So we actually have to link it back to the process and the effects. So here we are. Chromosomal mutations may involve large sections of the chromosome breaking off completely, or breaking off and reassembling in reverse order. So here we go. We are identifying those terms. Completely is deletion, inversion, breaking off, and adhering to another is translocation. These breakups of chromosomes may move genes to a new loci, and that's just moving them to a new location, and can break up genes by splitting the chromosome in the middle of the gene sequence. These types of chromosomes changes can radically affect cell activity. Chromosomal mutations also include non-disjunction of homologous chromosomes and anaphase. So we've talked about chromosomal mutations, and we've now talked about the mutations in the number of chromosomes as well. At anaphase, so here we are. There we go. So number of anaphase resulting the cell in many or too few. These can have radical effects on cell activity and the organism. And then we compare both point mutation and chromosomal mutation can cause disease. For example, point mutations can cause cystic fibrosis, sickle cell anemia, and chromosomal mutation can lead to Down syndrome and Turner's syndrome as well. Both kinds of mutations have also generated new alleles, which in some cases has been adaptive and contributed to evolution. So we kind of also want to talk about the fact that it's not always all negative. Make sure you know examples of each of point mutations and chromosomal mutations. Really, really important. Okay, let's have a look at biotech. How do genetic techniques affect the Earth's biodiversity? So in this section here, we're going to link it all back to genetic diversity. So biotechnology, we're going to start by looking at what are the social implications and ethical uses of biotech. We're going to look at what are the future directions of biotech, what are the potential benefits of society of actually using genetic technologies, and what are the possible changes that occur to the Earth's biodiversity as a use of genetic techniques. So this is our inquiry question: how do genes affect Earth's biodiversity? So let's begin by looking at what is biotechnology. So pretty much everything around us is biotechnology, right? Medicine, industry, food, energy, environmental sciences, computation, all of these things where we actually mix science and technology, that is biotech. And any technological application that uses biological systems and living organisms or derivatives thereof to make or modify products or processes for a specific use. So washing machine liquid, yogurt, because it's been fermented, butter or margarine, canola oil, all of these different elements around us, they are all produced through modification and the application of scientific procedures. So basically, biotech is technology based on biology. So let's talk about medical biotech first of all. We are all very familiar with them. It includes pharmaceuticals like vaccines and antibiotics, stem cell therapies. So stem cell therapies, for example, like skin grafts, the treatment of cancers and autoimmune diseases. That's also another example of medical biotech. So autoimmune diseases like treatment of type 1 diabetes using insulin created using penicillin. That's there. There we go. Bio-inspired materials like spray on skin, and finally diagnostics like biosensors, and we know how prominent biosensors are now in sports, and so on. Industrial biotech, it includes enzyme biotechnology, so improvement in enzyme biotech to actually improve the efficiency of industrial processes, includes biodegradable plastics and like production of biopolymers that are derived from plants and bacteria, and also energy sources including biofuels and photosynthesis. So the modification of these processes to try and produce different alternatives. Environmental biotech, so bioremediation is the use of microorganisms to clean up pollution, like algae, for example. And agricultural biotech is the biggest example is genetically modified organisms, so GMO crops that are, that are being used to lead to better yield, and also to be more suited to the changing environment. So what are the positives, right? I actually want you to think about this. What are the positives of using biotechs? Well, first of all, it meets the growing needs of society, right? Like, we are obviously growing population. Population is growing. Our resources are limited, and so by using these biotechnological products, we can meet the growing needs of a society. We can try and reduce that pressure on the environment, and we can improve quality of life through it. And that includes, for example, meeting the United Nations sustainable goals. So, like, ending poverty and hunger, improving health and well-being of individuals, access to clean water and sanitation, clean energy, sustainable cities, right? And so we are harnessing we're harnessing the power of biological tools to invent new creative solutions. In terms of genetic diversity as well, there is a positive because it leads to the creation of new arrangements of genes and new allele combinations. And it also allows for organisms to actually adapt to their environment. And ultimately, as a result of that, it can lead to the process of evolution being accelerated. And we now, and another big sort of part of this is open source information, which is a bit of a gray area here, but we can count it as a positive because it does lead to the creation of public databases, which allows for a growing amount of biological knowledge to actually be shared amongst people. Though there are concerns, and many at that. Firstly, ownership. Should we allow people or big corporations to actually own biological information? And there can be economic benefits, right? And that can lead to like the production of monopolies that possess biological processes, and the intellectual property constraints as well, so they can become prohibitive to process. So there is that line, between natural and synthetic that is being blurred. Commercial implementation is another one. Again, biotech monopolies, companies that have vast resources, that have vast finances to actually dominate the market, drive up the prices of the products, and that can then be detrimental to the people who need it the most. And there's that program dependency on these big firms. Consumer rights and choices is another big one, and we also want to consider regulation. So governments, can they strike a balance between safety and innovation? Biohacking is another one, and also eugenics. So, future directions of biotech. So there is continual improvement and refinement of current technologies, and creation of novel approaches to actually approaches to emerging issues. And there's that growing, I guess, what's the word here? There is that growing use of backyard biotech, of people actually experimenting, and so we know that synthetic biology is an interdisciplinary approach where we can have an engineering approach to creating biological tools. We can combine molecular biology, genetics, biophysics, computer engineering, and we can break down our understanding of biology into parts and then rework that to create new innovative solutions. And this is driven by like student-led competitions. The innovation is pretty significant. But how does it affect biodiversity? So positives are that crops may be insect resistant, and that means that we don't need to use harsh chemicals with it which impact the environment. Crops may also have increased stress resistance and productivity, and that can lead to us maximizing the use of restricted land. Also growing vegetation where there are harsh environmental pressures. So we know that we can try and grow vegetables in inside, you know, buildings like eco hubs and stuff. There is research into that's happening, and in fact it has already been implemented. And also there is a deliberate introduction of variation through this. However, there is the ability for GM crops to outcompete natural crops. There is also the establishment of monocultures, of just like having one type of species of a certain thing, because it's like it has like good environmental yield and it's cheap and inexpensive. So a good example of that is like Cavendish bananas, and so that can lead to the loss of genetic diversity. There's also horizontal gene transfer into native ecosystems, and competitive advantage can also lead to loss of naturally occurring alleles and variation. So when we evaluate this, and you should be able to evaluate, and you want to evaluate this, you know, in your own understanding, and with, I would recommend like having more examples to back up these points. Remember, I'm just, you know, outlining the content to you here. I definitely recommend going ahead and doing some more research, adding to these notes. So when we evaluate this, we can obviously say that agricultural practices, they are driven primarily by short-term economic profit, and they have posed a risk to biodiversity. However, biotechnology gives us the tools to enact this at a more rapid pace, and also at a more fundamental level, or a more genetic level. It does have a huge potential to help the agricultural industry, but it has to be used wisely, and with the consultation of necessary stakeholders. And that is evaluating the effect of biodiversity of using biotech in agriculture. Again, I'll skip these, but definitely recommend you go through it. I'm going to move on to the third part of this module. So here we are, genetic technologies. Does artificial manipulation of DNA have the potential to change populations forever? So here we go. We're going to discuss the uses and advantages of current genetic technologies. We're going to talk about reproductive technologies, cloning, recombinant DNA technologies, benefits to agricultural and medical and industrial applications, effect on biodiversity of agricultural biotech, influence of social, economic, and cultural contexts. And our inquiry question is, does artificial manipulation of DNA have the potential to change populations forever? So here we go, genetic technologies. This is what we are going to look at. Now, the key here is that some of them are listed specifically in the syllabus, but I'm going to talk you through them, and I just want you to know what it is, how it works, know examples for it. And I think even if you want to like draw down this little table, I think it's a really good visual of seeing actually what each one is. What is the reproductive technology, what is cloning techniques, and what are the recombinant DNA technologies that we're looking at. So let's begin by looking at artificial insemination and pollination. We've already talked about this in module 5. So artificial insemination is the injection of semen into the uterus without sexual intercourse. So we select animals that have favorable traits, and we basically take the egg and sperm, fertilize it, and we can also, what we can do is we can fertilize birth cycles. Sorry, we can synchronize birth cycles in order to essentially synchronize across however many number of organisms that are being produced, and it can also lead to reduced injuries. We then have in vitro fertilization. So in vitro fertilization, IVF, is when the egg is fertilized outside of the body. So so this is a fertility treatment that is used by humans to achieve successful pregnancy, and it also allows for embryo screening to occur. So here, for example, the process is that a woman is given hormone treatments to stimulate the production of eggs. So essentially, what happens is, once that happens, multiple eggs are taken from the woman's ovaries right there. In the labs, the eggs are mixed with a man's sperm, and they're cultured to become fertilized. Once they are fertilized, they're placed in an incubator for 48 hours. And then they're planted in the woman's uterus, or they're frozen for future implanting. Okay, here we go. So, reproductive technology is artificial pollination. So, plant sperm is purposefully taken from another plant, and it's placed in the stigma of another plant. So, if you want to know these terms again, it is where is it? It is a really good revision for Mod 5 as well. So it leads to the creation of new plants with favorable traits, so pure breeding, or we can also have crossbreeding, and genetic experiments, of course, like Mendel's experiment can also occur here. Now, putting it all together, here are the outcomes. Artificial insemination leads to the passing on of favorable traits and favorable genes to the offspring. Increased efficacy of livestock industry, however, it can lead to limited genetic variability. In vitro fertilization, able to reproduce with desired mate, it allows for genetic screening of embryos to avoid disease, however, it is expensive. And artificial pollination leads to higher crop yields, selection of desirable traits, and also to the creation of monocultures. Okay, another practice question. I've got the responses there for you. Have a look, have a go at these questions. It's a really good practice. Okay, let's now look at cloning. We're going to talk about whole organism and gene cloning. So, whole organism cloning, biggest example is the very famous Dolly the sheep. So it uses somatic cell nuclear transfer. So somatic cell nuclear transfer involves the removal of an adult cell from the organism that we want to clone. We then take an unfertilized egg from a donor and we denucleate it, so it's basically like an empty vessel. The denucleated egg and the stem cell are fused together, then the cell is cultured, it begins to divide. We then plant that embryo into a surrogate organism, and then the surrogate will give birth to an organism. Effectiveness is that offspring may not be strictly identical to the parent. There can be somatic cell mutations that can occur in the offspring. There can be mitochondrial mutations as well, because the mitochondrial has its own genetic information, and epigenetics is also part of it. It is an expensive process, and it's not time efficient, and it is better to just clone the organs of an animal rather than to clone the whole organism itself. Gene cloning. So, gene cloning, how do we do it? So, we essentially are creating lots of copies of a gene of interest. And, where is my, there we go, highlighter. Lots of copies of a specific gene, okay? So, essentially, we assemble recombinant DNA. So, we identify what the target gene is. We cut the gene out or order it online. So, we cut the gene out. We then treat that gene fragment and plasmid with restriction enzymes. So we treat it with restriction enzymes here, and what that means is we cut the gene in specific places, and then we create sticky ends of that gene. We then combine the sticky ends together, and once the sticky ends come together, they come together by base pairing affinity, and then we treat with DNA ligase to repair the backbone. The replicate, so that's the first bit there. We remember, DNA ligase should sound familiar from DNA replication, where the ligase joins together the Okazaki fragments. DNA replication, so after we've done that, we insert the recombinant plasmid into a host bacteria. Bacteria is used because they are super microfactories, like they can replicate really fast. So we essentially put that plasmid into the bacteria. It will express the gene and produce lots of proteins. And again, if you want to actually get a large number of the DNA copy, then we don't, and not the protein, because the DNA will produce the protein. But if you want to get the DNA copies itself, we can use polymerase chain reaction, which is basically going to spur like thousands of copies of the DNA. So here is the process. There's the gene, there's the plasmid. So essentially there's the plasmid, there's the DNA, we've got the bacteria, we've got a plasmid, which is a circular DNA plasmid. Here's the bacterial plasmid. We're going to put a restriction enzyme in it so that we can cut it open at a specific spot. We've got the linearized DNA with our target gene, and we're going to then put the two together right there, and that's going to lead to a recombinant plasmid. We're going to pop this into bacteria, and this has been used to produce insulin. So in the treatment of type 1 autoimmune disease, type 1 diabetes, where the person's body does not produce insulin, which means they can't regulate their blood glucose levels, and so that's where we can use this process to produce insulin and help them, yeah, essentially regulate their blood glucose levels, and as a result survive. So effectiveness is that it's fast, it's cheap, bacteria is easy to work with, and it is also easy to buy and sequence DNA. Okay, recombinant DNA technologies. Let's have a look at how we, this is essentially recombinant DNA technologies. It's a method to join together DNA from two different species in order to produce new genetic combinations. So transgenesis is the process through which we introduce exogenous genetic material, which is like material from outside the person, into another organism. And it is performed so that the organism can exhibit a new trait. And remember, DNA is ubiquitous, it's the same across organisms, so we can insert new genes into genomes. And the way we can do it is we can either do it through plasmids, retroviral vectors, or DNA microinjections, which is like a fine glass needle. So this can actually be done for agricultural and medical applications. So what do? We begin by identifying a gene of interest. We extract that gene right there. We take it and we're going to deliver it, and we're going to put it in a culture, and then we are going to lead to, and then we're going to grow it so that we can have a genetically modified crop right there. So how do we make a transgenic organism? The first thing we do is we identify our genes, then we get large amounts of a gene of interest, and then we insert the gene into a target organism. The first technique to get to identify our genes is gene sequencing. This is what we looked at DNA sequencing in module 5, so I'll just skip through this, but it allows us to gather single nucleotide information. We can identify genes with useful traits, and we can use that. The second step is basically using PCR. Either PCR or gel, either PCR and then gel electrophoresis. So PCR to amplify the DNA, to get lots of copy of the DNA, and then we're going to use gel electrophoresis to visualize the DNA to check that we have the right type. So we're going to get large amounts of it, but we're going to check that we have the right type, and then we're going to insert it into a target organism. We can do it through restriction enzyme cloning, so what we looked through in that earlier example, where we basically get the target gene, we get the plasmid, and we basically put it together, and we get a recombinant plasmid, and we're going to put it into a bacteria. We can also use transfection, which is basically we're using a virus as a vector. We're going to use a virus as a vector to put the gene into the target cell. Remember, viruses, they don't have their own machinery to produce proteins. They will go into what is going to happen is we're going to use the virus as a vector. So we're going to put a gene into the virus. We're going to send the virus into the target cell, and the virus is going to hijack the machinery of the target cell. It's going to then lead to the target cell. It's going to ask the target cell, basically force it to then make the proteins of that specific gene that we wanted to make, because that's what viruses do. But this time, instead of having the viral proteins in there, we're going to have the genes that we want. And finally, microinjection. We use a very fine needle, and we just, because the cells are small, and we just inject the DNA. Okay, here we go. That's the whole process. I will leave that for you to have a look at. Let's talk about some examples. So agricultural example, BT cotton. BT cotton, so basically the Bacillus thuringiensis produces Bt toxins, which are insecticidal, so they kill moths, beetles. And what we have done is the Bt toxic gene is inserted into cotton crops, because when pests eat the crop, the toxin is released and the pest is killed, and that reduces the need for farmers to actually use insecticides. The medical example of E. coli that we just talked about when we talked about recombinant DNA. So we know that insulin is an essential hormone which aids in the regulation of glucose metabolism, and inadequate production of glucose can lead to the development of diabetes. And what we do is we insert human insulin into E. coli, which is a bacteria, and we allow it to produce large amounts of it. We then take it, we purify that protein from the bacterial cells, and we use it to treat diabetes. Here we are. Recombinant DNA technology. So gene therapy is the correction of disorders. So we can actually correct disorders by introducing changes into the functional genes, and we can introduce healthy genetic material. The cell and its offspring can then have restored function, and we can insert using viruses, gene guns, or inorganic nanoparticles, and it can happen either in vivo in the living thing or ex vivo. CRISPR-Cas9 is a technology that actually edits the system for accurately introducing point mutations. So we introduce point mutations to fix something in the genome. So it has two parts. It has the guide RNA, which contains the nucleotide sequence, and it has the Cas9. The guide RNA is basically going to contain the complementary sequence that we want to edit, and the Cas9 is an endonuclease. It's actually going to cut the DNA so that the guide RNA can get in there and fix it up. ELISA is enzyme-linked immunosorbent assay. So what that is that it's a biochemistry tool which detects the presence of antigens in a liquid sample. So what it does is it's a diagnostic tool which exploits natural antigens antibody interactions. So within our bodies we have this antigen antibody interaction, and what happens is that the levels of biomolecules in a sample can be measured by linking levels of antigens or antibodies to a fluorescent readout, and it's useful for forensic epidemiology, and we can trace the spread of infectious diseases using this. So all our cells have antigens, and those are like our body antigens, like our self antigens, but every time we encounter a virus or a bacteria, they have their own antigen, and the body recognizes that as like an outside thing, right? Like it's a non-self particle, and then the body has its own antibodies that it uses to latch onto the antigens and identify those antigens as non-self. But these are the three right there. I would recommend watching like actual videos of how these technologies work. You can find some really good ones, because that will give you like a visual of how they work. I know we're going a bit over time here, so I'm going to finish up. I'll leave just practice questions for you. Last bit, here we are. So agricultural techniques and benefits. So techniques include selective breeding, artificial pollination, transgenesis. Benefits are that it leads to the creation of crops and livestock with favorable traits, high yields, high nutritional value, better temperament, and also reduced environmental impact. They can create organisms which do not require insecticides or herbicides, and it increases food security. In terms of medicine, there can be cloning, gene sequencing, gene therapy, CRISPR that we've looked through, and ELISA. Benefits are we can have personalized medicines. There can be preemptive diagnosis, treatment of genetic diseases, potential cures using like CRISPR-Cas9, and gene sequencing, improved diagnostic tools, cheaper, faster treatments, and creation of molecules which can be used for medical treatments like insulation for diabetes. Industry techniques, gene cloning, sequencing, transgenesis. So benefits are we can have increased speed of chemical reactions, more efficient industrial processes, creation of many everyday items, and creation of organisms which produce industrially significant products like biofuels, biomaterials, and energy. I'll leave the positives and negatives for you. They're very similar to what we have discussed. I will also leave the social, economic, and cultural context for you, because this is just putting together what is the influence of social, economic, and cultural context on biology, on these biotechnologies. So that includes, like looking at the big picture of countries, law, environment, religions, economies, and bringing that down to the science and the students and the relationship between the two. So that's there. These examples I will leave for you to have a look at, and this is what we have covered, and these are some resources for you to have a look at if you want to get some more examples for genetic technologies. Top tips: understand the fundamental technologies, know mutations, know examples, know your examples, and also memorize named examples. Do your research, really important. Check out those resources and be critical when you talk about the influences and the implications of biotech. And exam prep: consolidate your notes, really important. Don't get behind on them. Create module summaries, that can be super helpful. I personally found mind maps to be really helpful. And do practice papers, even if at the beginning it's just a couple of questions. Do it. Exam prep: mind maps, palm cards, really helpful. I personally found mind maps really good because it helped like really connect things together. Exam prep: when it comes to multi-choice, don't get caught up. Use process of elimination and be quick. Short answer question: pay attention to mark allocations. Use clear and concise term, and also define the key terms before you begin. Also, short answer questions: if you find anything that you are not sure about, move it, leave it, move on, and come back to it. It's a very underrated technique and a very underrated suggestion, but I think it always works really well. And long responses: use planning plus structure. Structure is really important for long responses. Don't forsake structure, and make sure your argument addresses the question. Again, these are some tips that I'll leave for you to read, and that is it from me today, guys. Sorry for going a bit over time, but I hope you found that helpful. Best of luck to you for this year. You're going to smash it. Have fun. Yeah, don't forget to have fun. Look after yourselves mentally and physically, and have a wonderful year. I hope I'll see you at the next lecture series. Take care, enjoy, and goodbye. Have a good one.